A lot of people are scared of statisticians. They're scared of giving a presentation, and afterwards, when they're up on the podium, the statistician comes to the microphone and they say, "Can you explain the multivariate analysis and the p-value?" And they get very worried, but I must say I really enjoy asking those questions. It's all part of the fun. So statistics can be your best friend and your worst enemy, and it's very easy to do statistical tests on your own and make mistakes. And if you do it properly, it works well. But what I'm going to go through this morning is all the things that can go wrong with statistics and how to avoid them. Lots of examples of HIV clinical trials and how to interpret them. So first of all, type 1 error means it's statistically significant, but you've already done 100 statistical tests. The more time you do a test, the more likely you are to find a result that looks significant. Now, a simple way of understanding this is if you throw 2 dice and you get a double 6, the probability of a double 6 is 0.028. It's 1 in 36. So that, if you think about the probability of something happening, the p-value is less than 0.05, remember a double 6. Things can happen by chance. Now, the most famous example of this is a beta blocker called atenolol. And the people who, there was a big clinical trial where there was a benefit of atenolol over placebo. And the authors submitted the results to the Lancet. And the review from the Lancet said, well, that's great, atenolol is better than placebo, but is it better in women or in older people or in white people? So the statistician for the trial, he said, well, those are all subgroup analyses, there could be type 1 error, so I'm going to do the subgroup analyses, but I'll also do some others. And he did an analysis of this study where he showed that atenolol was better than placebo only for people with the zodiac sign of Sagittarius. So it's a test, it was significant, is it true? So you imagine what would happen then. The patient comes into the clinic and you say, should you take atenolol? Well, your zodiac sign is Capricorn, so I don't think so. So you've just got to be careful. Some people, they'll do statistical analyses, they'll get results and they'll think, I know I've done lots of tests, but this one's true, this must be correct. So when you're doing an analysis, just think about how many tests you're running. And if you look at HIV clinical trials, we fall into this trap. If you look at, say, infuvitide, T20, on the FDA drug label, it says there's a risk of bacterial pneumonia if you take infuvitide. There were so many tests done on different safety endpoints. You wonder if pneumonia really is a possible adverse event, or is it just a result of doing lots of tests? If you look at the START Merck study, which was raltegravir against efavirenz, it was not significantly better at 48 weeks or 96 weeks, and Merck carried on doing more analyses, and I think it was eventually significantly better at 144 weeks or 192 weeks, and then people said, oh, now it's better, but they've done 4 tests. Similarly, Artemis, darunavir versus lopinavir, was not significantly better at 48 weeks or 96 weeks, and eventually later on it was significant, more and more tests. So you can get these situations where people keep testing, or maybe they'll do slightly different tests, and then they'll say, well, now it's significant, so now I believe it. But you've just kept doing things. And just remember, for type 1 error, 0.028 is like a double 6. There's nothing magical about a p-value less than 0.05. A clinical trial has a single primary efficacy parameter. That's how you judge the p-value. You can carry on doing lots and lots of tests, and you will get a significant p-value just by chance. So think about how that affects your analysis. One way to adjust for it is divide the p-value by the number of tests. So if you're doing 5 tests, you look for a p-value of less than 0.01. If you're doing it just carries on. If you're doing 50 tests, look for a p-value less than 0.001. Oh, I've already done that one. So the other thing you can get is you can get statistically significant values and differences that may or may not be clinically significant. Just because something is statistically significant doesn't necessarily mean it's important. I'll give you an example. So if you compare in the big phase 3 studies tenofovir, disoproxil fumarate, TDF, versus tenofovir alafenamide, TAF, you get a significantly better lipid profile for people who take tenofovir, the normal tenofovir versus TAF. You get significantly higher rises in LDL cholesterol for people who take TAF. Now, what's the clinical significance of that? Does that mean that more people will get myocardial infarction if they take TAF versus TDF? In order to do that, you need clinical endpoints. You would actually need to do a meta-analysis of all the studies of TAF versus TDF and look and see if that's true. In the same way, if you look at the clinical trials of TAF versus TDF, you get this significant change in eGFR. Now, and that's then in favor of TAF. TAF is better. So again, will this actually lead to more renal failure? Do we know that? If you look at the changes in bone mineral density for people taking TAF versus tenofovir, there is a statistically significant difference. But if you look at the START studies where people received either early treatment, mainly with tenofovir, or late treatment, including no tenofovir, there was actually no difference at all in bone fractures over 3 years in over 3,000 patients. So when you look at statistical significance, you don't necessarily know that it's clinically significant. And I think it's up to companies and researchers to do meta-analyses of proper clinical endpoints to validate any change that they see in these early markers. So the type 2 error is the opposite. You say there's no difference between the arms, but maybe the study was too small ever to show a difference. So we'll just sort of go back to basics. I'm not going to get you to do a sample size calculation. I'll just show you how it's done. So when you do a sample size calculation, when you design a study, the first thing is you need to know what is your expected outcome? What's your expected rate of something happening? And for that, you need to look at cohort studies and see how often things happen in real life. So let's imagine that we're setting up a trial, a clinical trial for a vaccine, preventative vaccine, and we want to lower the rate of HIV transmission by 70%, and we assume 80% power. Power means if you did the study 100 times, 80 times, it would show the result that you wanted. The 5% significance level means a p-value of 0.05. So first of all, you need data from a cohort study to see what your endpoint rate should be. So for a vaccine study, you need to know how often people are going to get infected with HIV. So here's the results from a paper we published in PLOS Medicine a few years ago. These are the rates of HIV transmission, the incidence, in lots of different populations. So if you look at people coming to sexually transmitted disease clinics in the United States, 1 in 200 or 0.5% get HIV every year. And the biggest risk group is Tanzanian police officers. I don't know what those Tanzanian police officers are doing, but 1 in 5 get HIV every year. So you can see you've got this whole range of different background rates. Now then, if you look at, on the y-axis is the sample size for the clinical trial, and on the x-axis is the HIV transmission rate per year, the incidence. And you can see, if you're trying to design a clinical study for a vaccine, you need to be absolutely sure about your incidence rate for that population. If you choose your assumed incidence rate too high, your trial's going to be far too small, and vice versa. So anytime you do a sample size calculation, it should normally be based on a systematic review of the endpoint rates in that population. Make sure to include as many cohort studies as possible. Now, the other thing about type 2 error is with HIV clinical trials now, we're getting very high rates of HIV RNA suppression. And so we're not doing difference trials. We're not trying to improve rates of virological suppression anymore. We're doing what's called non-inferiority. So a non-inferiority trial means we're trying to show that the new treatment is not any worse than the control, but maybe it has some benefits. So maybe that you've got two treatments that are the same in terms of virological efficacy, but the new treatment is once a day, or it has a better lipid profile, or it has a better bone profile, or a lower risk of drug resistance. So when you design a non-inferiority study, you have what's called a delta estimate or a non-inferiority estimate. So that means how much worse than control can the new treatment be, and you still say that it's acceptable. The other thing is the control treatment needs to be the recognized standard of care in that study and show a typical level of efficacy versus reference trials. I don't know how many people remember the BMS 034 study, which compared atazanavir and efavirenz. And unfortunately, Bristol Myers chose the wrong tubes to collect the blood in, which had the wrong anticoagulant. And their HIV RNA suppression rate was about 40%, way lower than you'd expect. And the European authorities did not accept BMS 034 as a study for registration. The response rate in the control arm was too low. So a little bit of statistics now. So let's say you've got two treatments. One treatment gives you 80% efficacy, the other one gives you 70% efficacy. Your difference is 10%. So your point estimate is minus 10%. And then you have confidence intervals around that point estimate. Now, in order for a trial to show non-inferiority, the lower confidence interval has to be above the delta or the non-inferiority margin. So this example here shows you where you fail non-inferiority. You're actually significantly worse than control. And in non-inferiority studies, you can get lots of different outcomes. You can be significantly worse, you can be neither better nor worse, but fail non-inferiority. The fourth one down, non-inferior, you have to be very close to zero actually to show non-inferiority, because your confidence intervals need to be quite tight, and then you can be superior to control. Now, here are some examples of studies where there's no significant difference between the arms, but they've failed to show non-inferiority. So if you take, for example, 2NN, a very famous study, very widely reported study comparing nevirapine with efavirenz first line. Now, in that study, nevirapine was 6% worse than efavirenz, and the lower confidence interval was minus 12.8%, which is lower than the non-inferiority margins of either 10 or 12%. So because of that, it fails non-inferiority. But at the same time, it's not significantly worse. So you could say this is like a type 2 error. You're saying there's no significant difference between the arms, but nevirapine is still not equivalent to efavirenz. In the same way, in MERIT, Maraviroc failed non-inferiority. It was 5% worse. The lower confidence interval was minus 11.2. Any study like this, you then cannot say that it's equivalent to the control. And interestingly, in the primary analysis of Gilead 903, which compared tenofovir to D4T, tenofovir failed non-inferiority. It was 4% worse, and the lower confidence interval was minus 10.4%. And so Gilead continued the study for another 3 years to show non-inferiority. Another problem you can get is you can get a company that runs a study and says there was no significant difference between the arms, therefore they're the same. You can't say that. You actually have to have the study of a large enough size. And normally, the big clinical trials in HIV that change practice have at least 300 patients per arm. Or you have a meta-analysis of different studies where the total number is more than 300 patients per arm. So this is an example. This is a study from ViiV, which is being enrolled at the moment for people who have HIV and tuberculosis co-infection, giving them either dolutegravir or efavirenz with rifampicin. The problem with rifampicin is it significantly lowers drug levels of dolutegravir. So you need to be sure that by doubling the dose, you can actually overcome that drug interaction. But it only has 60 patients per arm. So could you actually claim from that study that dolutegravir was better than efavirenz? Probably not. It probably needs to be larger. Okay, so type 2 error. Not significantly different does not mean the same. And a new treatment needs to be non-inferior to standard of care to be acceptable. And look at these 95% confidence intervals. The easy check to do is the lower 95% confidence interval above that non-inferiority margin? So if you look at that in terms of type 2 error, nevirapine is not significantly worse than efavirenz, but it's never shown equivalence. Abacavir is normally not significantly worse than tenofovir, but again, it's never shown this equivalence or non-inferiority. Okay, intent to treat analysis. The difference between intent to treat and as treated. Intent to treat includes everybody, and then per protocol or as treated analysis only includes people who are adherent to the study and haven't had major protocol violations. And the European regulatory authorities say that the results have to be consistent between an intent to treat analysis and a per protocol analysis. But if you think about intent to treat, I'll just show you some patients that I've worked on in my trials. So in intent to treat analysis, you include everybody in arm A if they were randomized to arm A. In arm B, everybody in arm B. So this is the first patient. They came into the Royal Free Hospital in London, and they were randomized in a study to 2 nucleoside analogues plus darunavir-ritonavir. And the patient said, I don't want to be in that arm, I want to be in the other arm. And so they stopped taking Truvada, and they took monotherapy for 3 years. They were randomized to triple drugs. They took 1 drug. But by intent to treat, you have to assume they took 3 drugs, but they didn't. Patient number 2. So this person, a drug addict, entered the study, took treatment for 2 weeks, cocaine for 2 weeks, treatment for 2 weeks, cocaine for 2 weeks. The viral load went up and down, and it's intent to treat, so you have to include them. We had people dying in car accidents. We had somebody who, in Poland, who came into the study and was put in prison for dealing heroin and remained in prison for 3 years, but you have to include them as a failure because they've been randomized. Now, so the problem with intent to treat is in real life with HIV, you get all sorts of patients who have all sorts of problems. And so what we do instead is per protocol analysis, we exclude those people and try and make sure that the study results are consistent if you include them or exclude them. So I'll give you a very simple example of a per protocol analysis. So in the 2NN study, if you look at the intent to treat analysis, including everybody who is randomized, there's no significant difference between nevirapine and efavirenz. In the 2NN study, if you just exclude people who are randomized but never treated, there is a significant difference between the arms. And what happened in 2NN is you got people who are randomized to efavirenz, and then they said, I don't want to take it. 19 people just were randomized, never treated. And in the Lancet publication, they were included as intent to treat failures. So just the most simple per protocol analysis can change the results of a study. Okay, next issue. With HIV clinical trials, we have these endpoints called missing equals failure, switch equals failure, FDA Snapshot analysis. And we need to remember it's a mix of virological failure and discontinuation. It's apples. In Britain, we have this saying, apples and oranges. We're trying to mix apples and oranges. And if you look at this is the classic endpoint now, it's called FDA Snapshot. When you look at any study produced by any company, it will say you're either a success at week 48, so your viral load is below 50 copies, or you're a failure. Now, you can be a failure because you have true virological failure, or because you've discontinued from the study, or you've had adverse events, or you've died, and we count them all the same. But the problem with FDA Snapshot is it just looks at a particular moment in time. You might have somebody who has under 50, 51 copies at week 48, and then under 50 again. That person's a failure because of that one moment in time. And also, if you look at the endpoint that we use in our HIV trials, if you look at the way that drugs are reported in FDA drug labels, only about a quarter to a third of the endpoints are actually virological failure. The rest is discontinuation for adverse events, but it's just classified as virological failure. Now, that's okay, provided that it's balanced between the treatment arms. So if you look at all of this data, this is a paper we published 10 years ago, looking at the percent of people in in drug labels, in the main clinical studies who had virological failure versus discontinuation for an adverse event versus discontinuation for other reasons, you can see that most failures in clinical trials, they're just people leaving studies. They're people, like I was saying, they're put in prison, they move house, they withdraw consent. But we're saying they're failures. Now, it's important to remember they're not virological failures, they're just people who've left the study. Now, let's take another example. So the Castle study, atazanavir versus lopinavir in treatment-naive patients, run by Bristol Myers, led to drug approval of atazanavir in Europe. Now, in the primary analysis, atazanavir is significantly better than lopinavir, 74% versus 68%, 6.1% difference. Confidence intervals don't overlap 0, so significantly better. You then do an on-treatment analysis, and it's not significantly better. What happened in Castle was lots of people left the lopinavir arm because they had toxicity, or because they just decided to leave the study. So if you look at pure virological endpoints, there's no difference between atazanavir and lopinavir. So it's all about toxicity. And then if you look at the single study, now some people think that dolutegravir is virologically superior to efavirenz. It isn't. It's exactly the same, if not maybe slightly worse. If you look at the single study and the percent of people who have virological failure, it's actually slightly larger in the dolutegravir arm than the efavirenz arm. 10% dolutegravir, 7% efavirenz. The difference between the arms is discontinuation of treatment, where 18% discontinued dolutegravir, 30% discontinued efavirenz. And that's why there's a significant difference between dolutegravir and efavirenz. If you look at drug resistance, there's actually no statistically significant difference between dolutegravir and efavirenz for the rate of emergence of drug resistance. It's not statistically significant. Some people say there's no resistance on dolutegravir, but if you look at the efavirenz arm, there was very little resistance there either. Another example is MERIT. MERIT was Maraviroc versus efavirenz in naive patients. So if you look at the primary analysis, it showed that Maraviroc was slightly worse, but not significantly worse. The overall analysis using FDA Snapshot was very similar. But then if you look at why people withdrew from the two arms, in the efavirenz arm, 14% of people withdrew because of adverse events and 4% for lack of efficacy. And you get the mirror image in the Maraviroc arm, where 4% withdrew for adverse events and 12% withdrew for lack of efficacy. So you get a situation where one arm is better tolerated but worse virologically, and the other arm is worse tolerated but better virologically. So that the problem with all our endpoints that we use in HIV clinical trials, they're this mix of apples and oranges, and you've got to make sure that the endpoints are well balanced between the treatment arms. Okay, and then when you look at virological failure, again, if we say that all of these endpoints are equal, I mean, obviously they're not. You know, in any clinical trial, people could fail with all sorts of different consequences. They could fail with high-level drug resistance. They could discontinue and switch straight onto an alternative treatment. They could go on holiday. They could stop taking their treatment. They could have a serious drug toxicity. And when we do analysis of trials, we just assume that all of these endpoints are the same. So it's important to look at them differently. So for example, if you look at the consequences of failing a boosted PI, if you look at a big systematic review we did, we looked at emergence of drug resistance. People who fail a boosted PI get very little resistance to nucleoside analogues, a little bit of M184V, and they're very unlikely to get a PI mutation. The same is true for integrase inhibitors. If you fail an NNRTI, you've got about a 50% chance of the M184V, a 1 in 3 chance of a non-nucleoside mutation, a 1 in 3 chance of an M184V, and then if you fail the same NNRTI treatment in a developing country where you have no diagnostics, you've got a very high chance of drug resistance. So the consequences of virological failure can differ between treatments. So here's another example where you actually have no difference between the arms. So this is tenofovir alafenamide versus tenofovir, the normal tenofovir. You've got no difference in RNA under 50 copies, no difference in virological failure, grade 3 or 4 clinical adverse events, or grade 3 or 4 laboratory abnormalities. So TAF and tenofovir have actually no difference in overall efficacy or safety, and that's probably why the German regulators have actually rejected the application to have tenofovir alafenamide included in the German health system. Then you lead, if you have no benefit, then you have a conversation about value. Is it actually worth including this drug in our formularies and paying more money than generic tenofovir in the future? Okay, so I'll just take you through a few individual cases, and we can look at when do you define virological failure using different algorithms. So the FDA Snapshot, it's all about week 48. It doesn't matter what happened before then. It just, if your viral load is below 50 at week 48, you're a success. TLOVR, the old way of analyzing data, you look at every value over time, and as soon as you have two values above 50, you're a failure. The ACTG, if you look at any ACTG study, it's about being above 200 copies twice. As soon as you're above 200 copies twice, you're a failure. So here's a classic virological failure. This is the SENT study of etravirine. So this person started off with 412,000 copies, remained at a high viral load despite treatment, and failed with high-level resistance. By any system, that's a virological failure. Okay, here's another one. This is somebody who went on holiday during the study, forgot to take their treatment, and developed HIV RNA of 40,500 copies. But you look at, imagine that analysis at week 96. Because the viral load is below 50, by FDA Snapshot, that's a success. It doesn't matter if they've had this time when the viral load's high. So the FDA would say that is a treatment success. The ACTG would say that's a failure because it's two viral loads above 200. There's one at 628 or 528, and there's one at 40,500. And then this one, so around week 48, two values below 50 copies. So the FDA Snapshot would say it's a success, ACTG would say it's a success, but the old TLOVR algorithm, because two viral loads are above 50 copies, that's a failure. So when you look at a paper published in The Lancet or AIDS, look at which system they're using to analyze virological failure, and you can actually get different results depending on which system they're using. As a general rule, the ACTG trials will give you about 5% higher response rates than company studies because their method is more generous, and it's harder to fail with ACTG than the other systems. So just some checks when you're reading a paper about a clinical trial. If somebody's claiming equivalent efficacy, is that actually true for the virological endpoints as well as the non-virological endpoints? So ViiV will say single shows that dolutegravir is significantly more efficacious than efavirenz. It isn't. There's actually no difference in virological endpoints between dolutegravir and efavirenz. Single shows that dolutegravir is significantly better tolerated than efavirenz, and that's a big difference. There's no statistically significant difference in resistance. If there was a difference between the arms, what was the reason for it? One treatment could be more effective, it could be better tolerated, or people might just stop one treatment arm more often than another. Remember, the ACTG has a different system of analyzing their endpoints, and you get response rates about 5% higher in ACTG studies compared to company studies. Okay, so cohorts, just a little bit on cohort studies. I know it's going to be covered elsewhere. Why would we believe the results of non-randomized cohort studies? Why not just look at randomized studies? Now, there's some reasons why we have to do cohort studies. Sometimes you just cannot randomize people to one intervention or another. And also, randomized studies often exclude a lot of patients, and you want to know what happens to people who are not included in studies. So if you look, for example, at recent claims that there are CNS toxicities on dolutegravir, there was some data presented at the HIV conference in Glasgow. Remember that the randomized studies of dolutegravir excluded people who had CNS problems at baseline. So we actually don't know what happens to dolutegravir when people have CNS abnormalities inside the main studies. We have to look at cohorts. There may be unresolved safety issues which can only be answered by long-term follow-up, either in cohort studies or meta-analysis of the clinical trials. If you look at the history of HIV, almost all the major toxicities we've seen have been after the phase 3 studies have been completed. Lactic acidosis, lipoatrophy, the cardiovascular toxicities of abacavir. There are many other examples, and we need the cohort studies to make sure that we have a good idea about safety. Another thing is sometimes you can't randomize. I'll just show you an analysis we've just completed looking at treatment cascades. So here on the y-axis, you've got the percentage of people on antiretroviral treatment. And on the x-axis, you've got the amount of military conflict going on in a country. Each bubble is a country, and the size of the bubble shows the size of the HIV epidemic. So on the top left, you've got these blue countries like Sweden, New Zealand, European countries that have a very stable, civilized system, very little military conflict, very high levels of antiretroviral treatment coverage. On the bottom right, you've got countries like Yemen, Somalia, Afghanistan, Democratic Republic of Congo, Nigeria, which have ongoing military conflict, and very, very low rates of ART coverage, about 3%, 5% in the worst countries. You could never randomize for this. You have to do a cohort study analysis and systematic reviews to actually establish this correlation. So in the real-life evaluation of treatments, remember that clinical trials often have strict inclusion criteria, and I've just shown you 4 studies down here where you can see the rate of screening failure. So people who should be entered into studies, but they have problems at baseline, maybe resistance, maybe CNS, and they can't get in. The other thing is that if you look at the number of patients you need to detect rare adverse events, you can get adverse events like attempted suicide on efavirenz, where you would actually need, you need tens of thousands of patients to show a significant difference. Right, well, I'm going to go through. I'm just going to show you a few. So this is my favorite example of why you've got to be careful about cohort studies. So this is an analysis of vitamin A cohort studies versus clinical trials. So there was a time in the 1970s and 1980s where people said, let's give everybody vitamin A. It's a powerful antioxidant, and it could prevent people developing cancer. And there were lots of cohort studies that showed the people who took vitamin A had significantly better survival than people who didn't take vitamin A. And there were, I think, 4 or 5 of these studies. So people said, well, let's do some clinical trials. And you can see on the top here, 6 different cohort studies showing most of them showing significantly improved survival for people taking vitamin A versus not taking vitamin A. And then the same treatment used in clinical trials actually made survival worse. It killed people. So you see this classic bias going through all the cohort studies. You imagine somebody who's taking multivitamins, they're probably less likely to smoke, they're not going to drink as much alcohol, they've got good exercise patterns, they're aware of their health, they're going to their doctor. So all those things are biases that are making survival better. And despite that, they're taking this vitamin A and they're still staying alive longer. That same treatment in clinical trials is making survival worse. So you've got to be very careful with cohort studies in believing that they're giving you the right result, because they can hide all sorts of things. People might just not say. People in a cohort study, if you say, oh, do you smoke? They say, no, I don't smoke. Then you just get the wrong information. You haven't got a randomization. So you've got an association in a cohort study, but you don't know if it's actually true. Another thing, you can get different results in different cohort studies. In the PEPFAR program in Zambia, tenofovir was associated with a 43% increase in mortality compared to cidovudine. It was actually worse for survival. And another study of tenofovir showed a decrease in mortality. Which one do you believe? I think people sometimes, they choose the cohort study that they want to believe, and they'll take the results from that one. But we've got to remember that cohorts can produce lots of different results, and I would always keep with randomized trials if you can. Now, I'm running out of time, so I'm just going to cut through a few things. Just finally, publication bias. Make sure that you get the whole picture. In the history of HIV, we have many trials that were never published, never presented, they just disappeared. And if you include those, it's quite hard to find them sometimes, but if you include those in systematic reviews, you can get different information. The best source of information on trials is either clinicaltrials.gov, which shows all the trials happening for a treatment, or also the European Summary of Product Characteristics that sometimes includes studies that were still never published. And this was never published, the study that showed efavirenz 200, 400, and 600 milligrams once daily all had the same efficacy. Despite that, for the last 20 years, we've been using efavirenz at 600 milligrams, and new studies have shown that 400 is just as efficacious but slightly better tolerated. It might be that 200 is as well, but if you don't see the full picture, you can't actually react. Levels of evidence, remember, when you're looking through guidelines, what's the grade of evidence that led to a treatment being recommended? Was it A1, supported by large prospective trials in a systematic review with a meta-analysis? Or was it B and C, which can include cohort studies down to expert opinion? This is a meta-analysis that we worked on, looking at 3TC and FTC, and the third line down, which showed that FTC was actually slightly worse than 3TC, was never published. And when we included that, it showed that 3TC and FTC were essentially equivalent in efficacy. And that's why the WHO recommends that you can either use 3TC or FTC. And similarly, a meta-analysis of PI plus 3TC versus PI plus 2 nucleosides, we're going to update this, shows no significant difference between the two. So just in conclusion, I'm trying to give you a sort of checklist that when you're going through a paper or seeing a presentation at a conference, what questions would you ask? What was the primary endpoint of the study? And what was the result of that endpoint versus lots of secondary endpoints? So when you see TAF versus tenofovir, remember there's no difference in efficacy, overall efficacy and safety, absolutely no difference between them. But you might be drawn to secondary analyses looking at bone markers or renal markers or maybe even lipids. What do they mean? Was the study randomized? If you get non-randomized studies, remember vitamin A. You can get all sorts of biases coming in. Was the study actually powered to show a difference? Is it big enough? What's the difference in virological endpoints versus all endpoints? So remember, the single study, there's no difference in virological failure between dolutegravir and efavirenz. Is the difference between treatments clinically meaningful? So you've got worse lipids for TAF versus tenofovir. Is that going to lead to more heart attacks? If you've got a difference in bone mineral density between TAF and tenofovir, is that actually going to lead to more bone fractures or no difference at all? Are you seeing all the evidence of a treatment, or are you just seeing some studies that are being highlighted by companies and other ones have not been published? And do you believe the results from a cohort study if randomized trials say something different? So I hope that's given you a not too hard introduction to statistics, and I'll answer any questions. Thank you.
I'm struggling a little bit with the differences you show, and you wonder why is there not like a true consensus within the major journals to publish the results in such a way that clinicians who are not statisticians can follow it, if you see what I mean. Because it's very difficult sometimes to follow your rules and then to sit with the paper. It can take you hours, and sometimes you won't even find the answers to your questions in the paper. So what explains the gap, if you see what I mean?
I think the problem is that the United States Food and Drug Administration have said to drug companies, you have to analyze studies in this way. Those are the rules, and whatever you do, we will analyze the studies in this way. So the FDA have the FDA Snapshot algorithm that goes into the FDA prescribing information, and if you Google, tenofovir or efavirenz or dolutegravir, you will always get the results in that way. The ACTG don't agree with them. They think you should analyze results in a different way. And other people say you shouldn't even look at switch equals failure, you should do switch included, you should look at the true intent to treat, the results at the end. But I agree, it's something that needs to be very clearly labelled so people can understand it. I think at the moment you're absolutely right, people can't.
We even have a setting that we have differences across the ocean, right? Maraviroc is being approved by the FDA for naives and not in Europe. Mm-hmm. So I think people like you should help us to clean up the process a little bit. But perhaps questions.
Sí. Andrew, first in Spanish. Es tu opinión sobre el margen de no inferioridad en los estudios de switch. It's your opinion about the non-inferiority margin in switch studies.
Yeah, so in a switch study, you expect a very high response rate. And some people say a non-inferiority margin of 10% is too much. It should be smaller. And I agree. I think with all of these studies, we need meta-analysis. We need to make these margins smaller, but when we combine all the studies. So if, for example, you look at PI plus 3TC, is that the right treatment to use? If you combine Olay with SALT, with Atlas, with Gardel, all these trials, we'll have 1,000 patients. So we can look at tighter confidence intervals. Yeah.
Over there.
One in the middle here.
Tengo dos preguntas. La primera es, ¿qué opinas sobre los estudios de comparaciones indirectas? So what do you think about indirect comparisons and how to extract conclusions in clinical practice from this type of comparisons?
I think you've got to be careful with indirect comparisons. I think that can be difficult to interpret because sometimes you have to take a difficult path from one drug to another. I mean, they're better than nothing. They're better than a cohort study, but they're not that reliable.
Y la otra pregunta es, cuando uno encuentra una diferencia estadísticamente significativa en un ensayo clínico que no ha planificado para ello, formalmente no podemos concluir diferencia estadísticamente significativa, pero me gustaría saber exactamente por qué no, y su utilidad para extrapolar datos. The other question is, when you claim for, or you find a statistically significant difference in a clinical trial, you have not pre-planned for that, for instance, in a non-inferiority study. What can we do with this finding? So can we take any assumption for that, or something is not valid at all?
Right. So there are two things really. The first thing is if you do a non-inferiority study, but one arm is significantly better, the European regulatory authorities, they do allow you to plan for it in the analysis, but you have to write it in the protocol that you were going to do that test. And I think most people now are writing it in the protocol. I know for the single study it was not in the protocol, and they had to negotiate to make that claim. The other thing is if you have a primary analysis of one endpoint, but then you look at lots of other endpoints, the thing to do then is divide the p-value by the number of tests. So if, say, you've done 50 tests, instead of p equals 0.05, you should look at p less than 0.001. If the difference is still statistically significant, then I think you can claim it. But if you get p equals 0.049 on a test after 50 tests, I would not believe that, because that's the type 1 error.
Andrew, es otra pregunta relativa a esto, a la afirmación de superioridad. Cuando hablamos de margen de no inferioridad, nos ponemos de acuerdo en una diferencia que es clínicamente no relevante. Pero en cambio, cuando hablamos de superioridad, no pedimos eso, es simplemente una diferencia estadísticamente significativa, hablamos. Entonces no sé si se debería hacer lo mismo, solo hablar de superioridad cuando todo el intervalo está fuera del margen de no inferioridad. You got the question.
Yeah, so it's statistical significance versus clinical significance. You can say, dolutegravir is significantly better than efavirenz by 7%. But what is 7%? Is that actually important? And I think then we need to look at what type of difference is it? You know, is it virological difference? No. Is it tolerability? You know, some people could argue, and actually if you look at the IAS guidelines for integrases, they're saying that if cost is an issue, start with generics, and if people don't tolerate them, switch to... So it translates. Yeah.
But the specific question, Andrew, is why don't we request that when claiming superiority, the whole confidence interval is outside the, let's say, plus minus 3% area of, you know, what we said?
Yeah, that would be, you'd have to have a very big difference, and we very rarely see those differences. I mean, normally you'll see a difference in a clinical trial of 6 or 7 or 8%, and then the lower confidence interval might be 1 or 2%, and I think that's not going to happen. Unfortunately, I wish it did, but it doesn't.
Yeah, first in Spanish. La pregunta va sobre las características que tienen que cumplir los estudios incluidos en meta-análisis. Regarding meta-analysis, you told us that they give us useful information, mainly when the individual trials are not with enough subjects inside.
Yeah.
So, but sometimes you are not sure that the clinical trials included in the meta-analysis are exactly with the same design. And it's quite difficult to analyze each one of these trials. For example, for simplification to boosted PI plus 3TC, if you are doing a meta-analysis with OLE, DUAL, and SALT, the individuals included in these studies are not exactly the same. They come from different treatments. So which are the key characteristics that we have to look for to assume that a meta-analysis is right, is well done? Which are the characteristics that have to share all the studies included?
So, I mean, the first thing is any good meta-analysis should be based on a systematic review. You need to be sure that you include everything. And then when you include all the studies, you do a test for what's called heterogeneity. It's called an I-squared statistic. And when you see the result for a meta-analysis, you need to be sure that, first of all, the baseline characteristics are similar between the studies. There's similar designs, similar endpoint protocol, but also that if you get one result in one study and then a very different result in another study, it makes it harder to believe the result of the overall meta-analysis. So you should get a p-value for the meta-analysis result and a p-value for heterogeneity. And both p-values need to be correct to believe the result. David?
A couple of questions. Should the primary endpoint for our large randomized trials be 48 weeks when it's long-term treatment? And then when you get 96-week and 144-week and you sort of carve it up beyond that? I mean, what is your opinion when you've got that kind of situation?
Okay, so the European regulatory authorities will only accept a drug for approval with 96-week results. And one problem with HIV clinical trials is drug companies, or people generally, they want to announce their results early. But if you announce the result at 48 weeks and you've still got people in the study, people might change their behavior when they see the result of the study. So they might see drug A is better tolerated than drug B, and they might think, oh, I might be taking drug B, I'm going to leave the trial. So really, studies should be run out to 96 weeks at least before you break the blind, before you do anything with the patients, because you could introduce bias otherwise. And I agree. I think, we're going to treat people for 20, 30 years. Why use 1 year of data? At least 2 years should be used, right? I think the studies should go on for longer. And actually, they should study everybody after they stop randomized treatment, because we can answer so much. I mean, for example, if you look at dolutegravir, there's apparently no drug resistance in the studies. But what about if you stop? Is there drug resistance longer? Does that mean you can start back on dolutegravir and get sustained suppression? So we need to study everybody out to 2 years and not just lose the patients when they stop randomized treatment.
And just comment on when you get non-inferiority but statistical superiority, that kind of situation.
Yeah.
So what's your view on that?
I don't see anything wrong with it, and as I said, the European regulatory authorities allow you to do 2 tests. The important thing is that the first test needs to be consistent for ITT and per protocol, because it's easy to show non-inferiority if everybody disobeys a protocol. It's called equivalence by default. You get lots of people doing bad things. But then the difference test needs to be significant for intent to treat, because it's more conservative. But there are statistical methods, and people are allowed to do that. Sometimes they say you've got to do 2 tests and adjust for multiple comparisons. So it's got to be significant at p less than 0.025 for the second test.
I have a question, but I reserve it for later because we're running a little bit out of time. Thank you very much, Andrew.